is Kristina, and I'm currently a PhD student studying in RNA bioinformatics. And today I'll be giving you a brief overview of what I do on a daily basis and how I use interaction networks to study relationships of RNA and proteins. So before we begin into the actual research, I first want to define what bioinformatics is. So the way I like to define bioinformatics is the integration of biology, computer science, and statistics. So why these three fields? So first we use biological data. So it can be data on DNA, protein, RNA, or even clinical data. And then we use a large amount of these data and then process them and analyze them using different computer science techniques such as machine learning, any data science techniques, and pattern recognition, and etc. And then the last question we want to answer is our findings really significant, and like how reliable are they? So we apply statistical methods to study and answer these questions. So in short, the way I like to define bioinformatics is applying data-driven decision-making processes to answer complex biological questions. So to give you some examples in different avenues in bioinformatics, first we have structure modeling. So what would the 3D structure of a protein or an RNA look like? There's also personalized medicine and drug discovery. So we apply bioinformatics to identify certain biomarkers. There's also clinical bioinformatics, so analyzing MRI scans or health and patient records, and applying bioinformatics to better characterize these images and records. There's also systems biology. So biology, it often ends up being complex biological systems. So we use networks or other modeling techniques to study these fields. And lastly, there's bioinformatics tools and database development. So we work with large amount of data, so it's important for us to keep track of these data sets and then update them on a regular basis so that we don't rely on outdated data sets. So what I do is the intersection of system biology and managing bioinformatics tools and database development. So I study RNA and protein interactions using networks, and specifically on SNRNAs, which stands for small nucleolar RNAs. Since most of you are STEM here, I believe, I think you've learned about the central dogma back in high school. So how DNA goes to RNA and then protein. Just to give you a refresher, I'm just going to give you a quick, easy analogy. So imagine you're at a bakery, and then of course there's going to be like a certain recipe book that's going to be followed. And then the baker decides to make a muffin that day. So he takes out the recipe of a muffin and then uses that recipe to create a delicious muffin. So if you link it back to a biological context, the recipe book would be the DNA. So that would be a book of biological or genetic information. And then the specific recipe for a muffin, that would be a strand of RNA. And then the baker would jump in and then use the recipe to make the muffin. And that's the role of the ribosome, so translating RNA into a specific protein. And then one day the baker decides to try something new, and then he decides to bake a croissant. But he does not have the expertise. So he decides to reach out to a croissant specialist, which would be the role of a SNRNA. So what the SNRNA does is it guides the ribosome to make chemical modifications on the RNA. So if we go back to the bakery analogy, the baker would need help from the croissant specialist to modify its recipe to make the perfect croissant. But the thing is, the croissant specialist, it also needs the best ingredients. So it reaches out to the experts of like specific butter and flour experts. So they really communicate together to create the perfect recipe together. And if you link back to a biological context, that would be SNRNAs and proteins working together to create the perfect protein product. So yeah, the goal is collaborative work of SNRNAs and proteins. But since this example was specific to a croissant, what if the baker wants to make different bakery goods, but he wants like each baked good to be a perfect product? Then he would need different teams of specialists and experts to be involved for each type of baked items. And if we translate this into a biological context, that will be different teams of SNRNAs and proteins making different groups being responsible for the creation of different protein products. So the hypothesis or the goal of my research project is that SNRNAs interact with different protein groups, and then by forming different groups, they are involved in different functional roles, having different regulatory impacts on their target molecules. And because there are so many different ways they can interact, the approach that I decided to use is to use an interaction network so that we could easily visualize complex interactions. So what I do is I combine or integrate different SNRNA and protein interaction data sets, and then I use an open source network visualization software called Cytoscape to overall create a network. And this is the network that I have, which is called Snowflake, standing for SNRNA Protein Functional Interaction Network Models, which is the one you see on the left. It's a pretty interesting network, and the dots on the outer circle, they represent SNRNAs and proteins. And then the lines, or edges you call it, that you see within the circle, they represent different interactions among the SNRNAs and the proteins. So if there's a line between two dots, that means the two dots are interacting. So what do we do with this large network? The network itself, it doesn't tell you that much, but what we do next is we kind of break down the network to identify potential subnetworks or potential connected hubs and identify repeated patterns within the network. So these are some examples of the patterns that I identified within this network. So to give you a better example of what we do further, so this is one of the most identified patterns within the network. And then what we do next is we look at their targets. So if these SNRNAs and proteins work together, what kind of functional effect would they have on their targets? And to answer this question, we work with wet labs because we have certain limitations with our computational methods, and it's important in biology that what we predict computationally is also validated in the wet lab experiments. So we looked at what if one of the proteins are not there, what kind of functional effect would it have on its targets? And in the end, we actually identified several genes that are involved in different diseases, such as Alzheimer's and PWS, and also cancer. So this tells us why it's important to study SNRNA and protein interactions. And eventually what we want to do is come up with a potential SNRNA and protein model, a more of a complex, and like how these models are formed and then study their functional effect. So to summarize how we went from the initial data set to the network and then the final model, we first integrate different data sets, construct a network, break it down, I try to identify different patterns, and lastly come up with a potential biological model. So why is this network important for different groups of people? So for RNA researchers, well, it's important for us because this network itself contains a lot of information, and overall we want to host this network in a web server so that it'll be available for different audiences, including those who do not have any programming experiences. And so far there hasn't been any interaction networks that involve SNRNAs because these SNRNAs are pretty new. Like they're a new type of RNA that has been discovered in the past few decades. For the general population, why would this be important for you? I'm not going to say SNRNA is going to cure cancer, but it does show involvement in different cancers and diseases. So our study in SNRNAs could contribute to new disease treatments and therapies. And lastly, as a CKC participant, why would this be important for you? So I introduced a new tool, Cytoscape, and how you can use network biology to study different interactions. So I think it's a useful skill to have or to know if you ever decide to work on different interactions, even if they're not biological. And yeah, that's what I do for my research. And yeah, thank you for listening.